Hello, I'm Dr. Terry Goss-Kinsey, the Interim Senior Associate Dean for Research here at Robert Wood Johnson Medical School, and it's my pleasure on behalf of Dean Amenta, Dean of Robert Wood Johnson Medical School, to welcome you to a very special event here at Robert Wood Johnson Medical School, the Kenneth S. and Audrey S. Gould Lecture. This lecture is a real highlight of our year in bringing together scientists, clinicians, students, and faculty, the Goulds and their friends who are here to really support the academic mission here on our wonderful children's academic campus here at Robert Wood Johnson Medical School. I want to particularly thank the Goulds for their generous support of this series, but also for helping to be at the forefront of building this real initiative on this campus in pediatric care, and especially bringing together researchers from both the basic, translational, and clinical sciences to address the tough problems in science. It's really been a wonderful catalyst for many opportunities for research here on our campus. And in fact, I just want to say that this year, Robert Wood Johnson hosted the university-wide research symposium. We used this very room for part of our symposium, which was focused on child health. So we're really greatly appreciate the stimulus this has been for this initiative here on our campus. And so with that, I'd like to introduce Dr. Arnold Rabson, the director of the Child Health Institute, to start our seminar. Thank you, Arnold.
Thanks, Terry, and I want to start with some thanks as well. First, again and foremost, thanks to the Gould family, to Audrey and Kenneth and the whole family for your really steadfast support, not only of this lecture series, but of the whole initiative of the Child Health Institute. You were there at the very beginning helping get this going. You've had the patience and the foresight and the generosity to help see it through, and I think with our 10 faculty now, we really have a very active, vibrant atmosphere here, and we're very grateful for your support. I want to thank particularly for this lecture, the Dean's Office, particularly Jeanette Evans and Patty Hansen, as well as Anne-Marie Koch of the Child Health Institute, who did a lot of logistical work to make this happen, and it's not insignificant. I'm very, very grateful for that. And finally, I want to thank all of you for attending and really supporting this activity that does stimulate the kinds of basic research, translational research interactions that we're really trying to promote here, particularly in the area of child health, and really bridge the gap of Piscataway basic science and basic and clinical translational sciences here. It's a tremendous pleasure for me to introduce our speaker for this year's Gould's lecture, Dr. James Downing. And Dr. Downing's accomplishments and titles are listed in the program, so you can see them all, but I'll just highlight a couple of features. Dr. Downing really, like myself, I feel a real affinity, is a pathologist by training who's become essentially a molecular biologist in his career and his contributions to the world. Dr. Downing went to medical school at University of Michigan and did pathology training at Washington University at a time when it was really the sort of hub and mecca, if you will, of anatomic and clinical pathology in the academic world with some of the real greats of surgical pathology. He then did hematopathology fellowship training down in Florida and ended up at St. Jude's, where, as described in the program, he really has risen through the ranks through the years to the point now where he's scientific director, executive vice president, and deputy director of St. Jude's, as well as the Gignac Chair of Pathology. I want to take 1 second, though, to highlight the science, because this is where I first really became familiar with Dr. Downing, and work that started a theme back in postdoctoral work with Chuck Shear looking at oncogenes stimulating the growth of hematopoietic cells, and really continued as he developed his own lab, integrating his clinical interests in leukemias and in the molecular pathology and molecular diagnosis of leukemias with an understanding of basic aspects of leukemogenic genes, particularly translocations involving AML1, the AML ito, TEL, AML translocations, so important in pediatric leukemias. And one of the really impressive things in looking at the kinds of work that Dr. Downing has done is he was one of the first people to use knock-in mice as a way to really characterize the functions of these translocations and how they end up causing leukemias in kids. So really a foresighted and really quite advanced use of a technology that in the early 1990s was just coming into real practice, common practice, to study these diseases. More recently, Dr. Downing again was a leader in implementing the gene expression analyses approaches to look at the different forms of pediatric leukemias, and as I'll talk to you about today, has done extraordinarily exciting work now looking at whole genome sequencing in these leukemias. So I think our understanding of how you get leukemia, how kids get leukemia, has just taken enormous leaps based on the work that he's done. So without any further discussion or taking more of your time, I'll introduce Dr. Downing, who'll talk to us about the molecular pathology of acute leukemia. Jim, thanks so much for coming.
Okay, well, thank you very much for the invitation to give this seminar. It really is a distinct honor, and I thank you for that invitation. And thanks for the great introduction. You know, it makes me stop and think for a moment. You know, I guess as you said, at one point in my career, I was a hematopathologist, and then I sort of became a mouse geneticist, and then back to molecular genetics, and now more genomics and bioinformatics, and I'm hoping ultimately you'll see to make it back into that diagnostic realm and actually where the information I generate affects patients. So what I'm going to do is sort of tell you a story of the data that we've been generating over about the last 7 years. And it's focused on acute leukemia. And this is a picture of pure leukemic cells that were taken from a little infant with acute lymphoblastic leukemia. And so acute leukemia is this proliferation of blasts that arise within the bone marrow or within the thymus, rapidly disseminate throughout the body, and if left untreated, lead to the rapid death of the patient. These are probably some of the largest tumors that we see in patients. I mean, think of this. This is just pure leukemic cells circulating in that infant. Kilograms of cells in an infant that doesn't weigh much more than that. Now, that helps us as researchers. We get pure cancer cells and we can look at it, but clearly a major burden for the patients. Now, acute lymphoblastic leukemia, the most common malignancy seen in the pediatric population, is clearly one of the great success stories of modern chemotherapy. If I had come down with acute lymphoblastic leukemia, I would have had less than a 5% chance for a cure. Today, if a child comes into the hospital with acute lymphoblastic leukemia, greater than a 90% chance for a cure. This is from different eras of treatment. And so in a single generation, we've taken what was almost a uniformly lethal disease and turned it into one of the most treatable forms of cancer that we see. So if one reflects on that and asks, well, how was that achieved? What were the fundamental observations that allowed us to make that progress? And they really are quite simple. It turns out the drugs we used to cure these kids existed back here. They were all there. We didn't know how to use them. We didn't know what combinations. So it was experimental protocols that designed how best to use those combinations of drugs. We didn't know how to support the children. We treat them with those chemotherapeutic agents, their white blood cells disappear, their platelets go down, and they would die from infections. So supportive care was required. But I would say that the progress really from about here up is really understanding risk stratification. That is that acute lymphoblastic leukemia is not a single disease, that there are different genetic subtypes of acute lymphoblastic leukemia. Some respond well to chemotherapy, others respond horribly. And so knowing which ones are the bad responders and treating them more aggressively. Not with targeted agents, by and large, but with standard chemotherapy, but more aggressive forms of chemotherapy or bone marrow transplantation. And so it really is this risk stratification that's allowed us to push to there. So what is leukemia? Well, it is this transformation of cells in the normal hematopoietic system. This is an exceedingly complex system. This arises from the hematopoietic stem cell. There are these multipotential progenitors. They differentiate into T lymphocytes, B lymphocytes, a variety of different kinds of white cells, platelets, red blood cells, an exquisitely regulated system. If you cut yourself, platelets go there. If you lose lots of blood, new red blood cells are produced. If you have an infection, white cells are produced. If you have an allergic reaction, other kinds of cells might be produced. So an exquisitely regulated process that comes and goes depending on the needs of the individual. But leukemia, what it is that we end up with mutations. Mutations accumulate in individual cells arising in that hematopoietic stem cell or a lineage-committed progenitor, and one or two mutations, not enough to generate a leukemia. In fact, most of those mutations are deleterious and the cell will disappear. But ultimately, if you have enough accumulation of mutations that don't kill the cell, but somehow give it some advantage, you'll end up with the leukemia. So what are the mutations? What do they do? What does it take to convert that hematopoietic stem cell into a leukemia? Well, there's a large list of different kinds of mutations, from point mutations in individual genes to deletions to rearrangements, inversions, amplifications, epigenetic changes, germline polymorphisms. So lots of subtle changes in genes and lots of major changes in genes may be the mutations that drive cancer. So if we want to know that landscape of mutations that drive a cancer, we're going to have approaches that allow us to detect all of these alterations. Now, I like to put this into some kind of context. I like to think, okay, if we're taking that hematopoietic stem cell, what biologic processes really need to be altered to convert that cell into a leukemic cell? What biologic processes do these mutations need to affect? And so I like to reduce it into a relatively simple model, and I like to say that there really are 4 biologic processes that must be altered to convert a hematopoietic stem cell or lineage-committed progenitor into a leukemia. You first need a mutation that somehow alters the self-renewal properties of that cell. Self-renewal, the ability to replicate itself without changing, is a property of the hematopoietic stem cell. In leukemias, we have lineage-committed cells. Normal lineage-committed cells do not have the capacity to self-renew. So you need some mutation that essentially allows the acquisition of that stem cell property into that committed progenitor. Second, you need an altered response to growth factor signals. Normal cells are controlled by homeostatic mechanisms. Leukemia cells are not. So some kind of mutation that allows those cells to proliferate in that uncontrolled fashion. You need a block in differentiation. Acute leukemia is a leukemia where the cells don't terminally differentiate. So some kind of mutation to block that differentiation. And so if you have mutations in these 3 pathways, it's not sufficient to generate a leukemia. In fact, that cell will self-destruct. It will undergo apoptosis, a process of cell death. And so ultimately, you also need to block that pathway to generate a cell that now can serve as that initiating cell for the development of that leukemia. So it turns out from many, many years of work of hematopathologists, cytogeneticists, we've learned a lot about the different genetic lesions in pediatric acute lymphoblastic leukemia and acute myeloid leukemia. As Arnold alluded to, many of us have been working on those translocations, doing all kinds of modeling of the biologic consequences of those translocations. Making knock-in mice where we knock in a translocation into a mouse, ask what's the effect of that on normal hematopoiesis, a variety of different experimental approaches. From that, what we've learned is those chromosomal translocations are initiating lesions. They start the process, but they are insufficient on their own to generate the leukemia. What we do know is that they do fit into particular of these biologic processes. We know translocations like the 821 that generates an AML1 EDO fusion, or translocations involving the MLL gene, by and large, they alter the self-renewal property of the cell that they're expressing it in. Others, like translocation 922 that generates a BCR-ABL fusion, this is an activated protein kinase, tyrosine protein kinase, generates a growth factor signaling process that induces proliferation and enhances survival. RAR alpha from acute promyelocytic leukemia, it blocks differentiation. So we are learning about these, but we know they're insufficient by themselves to generate leukemia. So the key question that we've been focused on, and many people in the field have been focused on over the last 10 years, is what is that total complement of mutations required to alter all these pathways? What does that complement mutation tell us about the biology of that leukemia? Can that information be used to help us better diagnose leukemia, perhaps better risk stratify leukemia? And ultimately, if we knew that landscape of mutations, could we better treat the leukemia? So we're focused on defining what is the landscape of mutations in pediatric acute lymphoblastic leukemia. But as you'll see, we've expanded to acute myeloid leukemia and then really to all pediatric cancers. Can we understand that entire landscape of mutations? So let me just start with a study that's a bit dated now. It was published in 2007 in Nature, but it at least gives you the thought process and the way we approach this. And what we've done is move with the technology over the last several years to approach this specific question. So what we're doing then is we're taking leukemic cells from the patient at the time of diagnosis. We're doing routine diagnostics, routine hematopathology diagnostic, flow cytometry. We're doing molecular cytogenetics and FISH analysis. But then we're extracting DNA and RNA from the leukemic blast, and then using genome-wide approaches, first to look for copy number alterations using the Affymetric single nucleotide polymorphism arrays, and looking for gene expression using a variety of different platforms from Affymetrics, interpreting this data, and then going back and sequencing individual genes that might be dysregulated or were predicted to be dysregulated from that data, and then integrating this data using computers to help us identify new biologic markers of potential risk, new markers of potential therapeutic targets, and insights into the biology. So the first study we did was a study using 242 diagnostic leukemia samples from pediatric patients. We had DNA from the leukemic blast, and we had matched non-tumor germline DNA from each patient. And what we're going to do is single nucleotide polymorphism arrays, look across the genome at about a 5 kb resolution to look for copy number alterations. Are there focal amplifications or focal deletions that are occurring somatically in the leukemic cells? So pretty straightforward experiment. That experiment cost $1 million 6 years ago. So, not the kind of thing you can write an R01 for. Nobody's going to give you that kind of money to do this kind of experiment. In fact, as we discussed it around the institution, I'd say more than half of the people said that's crazy, that you're not going to get anything out of this experiment. And the reason you're not going to get anything out of this experiment is cancer's too complex, and you're going to see lots of alterations, and you're not going to be able to make sense of it. I'd bet the other half said, well, I'm not sure you're crazy, I just think you're stupid, because in fact you're not going to find anything, that there just aren't going to be any hallmarks of the genetic lesions you're looking for. It's complex, and you're not going to get sort of the smoking gun that's going to give you biologic or clinical insights. So of course we said thanks a lot, we got the money, we ended up doing the experiment, and it was, probably when I saw the results, I immediately knew it was the most important results I'd ever seen. And so what we found was first that there weren't that many genetic alterations. It's a small number of copy number alterations, amplifications and deletions. Now we had great molecular diagnostics on these cases, and so we found that the number of mutations or number of copy number alterations varied markedly across the different genetic subtypes of ALL. So one subtype is infant acute lymphoblastic leukemia. It has a rearrangement of the MLL gene, and it had, on average, less than a single copy number alteration per case. So that says very few cooperating mutations. You've got this MLL rearrangement and very few cooperating mutations. Makes sense. The latency for the development of that leukemia is very short. The MLL rearrangement occurs in utero, and sometime during that first 12 months, the patient develops overt leukemia that takes over the body. At the other end were hyperdiploid leukemias, a good subtype of leukemia, but they have trisomies of many chromosomes, and so, of course, they had many lesions, whole chromosome gains. The second surprise was that deletions were more common than amplifications. If I were a betting man, I would have bet amplifications were going to be more common. Amplifications lead to overexpression of genes. One would predict we would see overexpression of oncogenes driving the leukemia. No, what we saw were deletions. Turns out those deletions are actually a result of aberrant RAG-mediated recombination that is the normal process generating antigen receptor rearrangements in the cell, and somehow it leads to focal deletions, and if those give a selective advantage, they're selected as part of the leukemic cell. The most surprising result is this, though. 60% of B lineage ALL have a genetic lesion in a gene regulating normal B-cell differentiation. Made perfect sense. Here's normal B-cell differentiation from a stem cell to a lymphoid precursor to pro-B, pre-B, pre-B, and to mature B. And these genes were PAX5, a transcription factor that controls that stage of differentiation. EBF1 and E2A that controls this step, and Ikaros, which controls a very early step. In fact, the surprise was that nobody had looked at these previously. Nobody had looked at these in pediatric acute lymphoblastic leukemia for mutations because they hadn't really been thinking deep enough, us included. But from this genome-wide scan, what we see is 60% have mutations, with the highest gene being mutated is PAX5. So let me just show you what some of that data looks like. So here is the PAX5 gene. It is a transcription factor. It has a paired DNA binding domain. It has transcriptional regulatory domains. It's encoded by 10 exons spread over about 200 kb of genomic DNA. And what I'm showing you here is the extent of deletion in individual patients, each line with a solid line being monoallelic deletion, deletion of one allele, a dotted line being biallelic deletion, and red being an internal amplification. So about half of the cases delete one complete allele. They have one normal allele, the other allele is deleted. If you look in those leukemic cells, it's true haploinsufficiency, half a dose of PAX5. The other ones, though, were internal deletions, and many of these you could actually splice across the deleted segment and actually make an internally deleted form of PAX5 that was expressed in the leukemic cells and lost either DNA binding domain or transcriptional regulatory domain. So a hypomorphic form of PAX5. Reduce transcriptional activity. Several had deletions that were only at the 3' end of the gene. Those were reminiscent of chromosomal translocations. Often in translocations, you lose components of the DNA at the junction. So we thought, could these be translocations? And went on to prove they are translocation. This is FISH showing, in this case, it's a fusion between PAX5 and ETV6. Here, PAX5, FOXP1. Here, PAX5, ZNF521, which is EVI3. And it turns out these fusions, each of the partner genes in the fusion plays a normal role in lymphoid development or has been implicated in leukemogenesis. So these are two hits in a single chromosomal translocation. And then we went on to sequence PAX5, and we found point mutations in PAX5 that disrupt DNA binding or transcriptional activity. So overwhelming genetic evidence that these alterations are playing a role in contributing to the development of acute lymphoblastic leukemia. And it makes sense. Remember, again, the ALL is blocked in differentiation, and so these lesions are contributing to that block in differentiation. But it's interesting. It's loss of one allele. It's never complete loss of PAX5. It's really as though if PAX5 were a transcription program pushing differentiation, what we've done is like a rheostat, just turned it down and helped to block that process of differentiation. So this was great data. We were excited. I'm convinced that these were involved. But being a mouse geneticist, started thinking, well, wait a minute, what does the mouse look like if I knock out one allele of PAX5? Do they get leukemia? Because I'm saying loss of one allele must be contributing to leukemia. And so if you look at the literature, if you lose one allele of PAX5 in a mouse, the B cells are perfectly normal. They don't block differentiation. They don't even slow down differentiation the way people looked at it. And there's no evidence of leukemia spontaneously developing in those mice. So does that mean this is wrong? We again looked at that data and said, well, you know, they actually haven't done the experiment. Nobody's really done the experiment. And so I'll just show you one piece of it. But what we did was we took bone marrow from either mice that are wild type for PAX5 or were heterozygous for a null allele of PAX5, so theoretically haploinsufficient for PAX5. Transduced the bone marrow with the retrovirus expressing BCR-ABL off an internal ribosomal entry site, so driven at a low level. This is a relevant model because 50% of BCR-ABL ALLs actually have a PAX5 deletion. And we asked, does the loss of PAX5 accelerate the onset of leukemia in this model? So now we're asking, in the model, we don't get spontaneous leukemia, but does that loss of the allele actually accelerate the onset of leukemia, cooperate in leukemogenesis? And the answer was yes. You can see it increases penetrance, decreases the latency of the development of the leukemia, and if we program in EBF loss or Ikaros loss, or the combination of the loss, we see further progression of the acceleration of disease onset. I should also point out that although B cells look normal before we put in BCR-ABL, if you stress the B cells, they have abnormalities. They don't differentiate quite normally. So the loss of these genes is a direct collaborator in the process of leukemogenesis in pediatric ALL. So we went on from there and did a number of studies that have all been published. So I won't go through in any detail. But we looked also that the deletion or mutations of Ikaros, it turns out, were near obligate events in BCR-ABL positive ALL. We went on to show that, in fact, in BCR-ABL negative ALL, a mutation in Ikaros is a very bad prognostic indicator. That if you have that mutation in Ikaros, very aggressive form of leukemia and probably needs to be treated differently. We pursued that in a little more depth, and it turns out that these cases, BCR-ABL negative that have Ikaros mutant, they actually have activating mutations in JAK family kinases. They have rearrangements of CRLF2, and more recently, just out in Cancer Cell, they have those that lack JAK mutations actually have rearrangements that generate fusions of other kinases. And so this is a highly aggressive form. We can identify it by the presence of the Ikaros deletion or mutation, and they're responsive to inhibitors of these activated kinases. And so again, maybe this needs to be incorporated upfront into risk stratification and actually targeted therapy. So genome-wide screen, a lot of information. Information about the basic biology, PAX, EBF, Ikaros, a risk stratifying lesion, Ikaros deletion or mutation, and therapeutic targets, JAK activating mutations, CRLF rearrangements, and these fusions that can include JAK or PDGF receptor or a variety of other kinases. So we were quite excited by this data. We were excited to continue and to look deeper. And so we knew there were large sequencing projects being done around the country. We knew the Cancer Genome Atlas project. The federal government was spending somewhere between $100 and $300 million in doing sequencing of adult cancers. A very small portion of that, a couple $1 million, was being focused on pediatric cancer. And we were participating in those studies with most of the good data coming out on acute leukemia through a cooperation with the COG. But we felt what needed to be done was somebody step up and sequence pediatric cancer. Why not do a pediatric cancer genome project? So about a little over 3 years ago, we got together and said, well, what would it take? What would it look like? How could it be done? And so we spent a year really planning a pediatric cancer genome project. We had faculty retreats. We had open discussions at those retreats. We purposely solicited faculty members to say why we shouldn't do it. We solicited other faculty members to say, well, here's what we would do if we were going to do it. And so after a thorough process with support from our Board of Governors, we decided let's go forward with the Pediatric Cancer Genome Project. We decided that we would partner with one of the top 3 genome centers in the world at Washington University, just 5 hours up the river. And so we set out a project that was a cooperation between St. Jude and Washington University Genome Center to sequence 600 pediatric cancers and match normal DNA. And this was whole genome sequencing. And to do this in a 3-year period at a cost of $65 million. And to completely fund it privately, not a cent from federal dollars. And in fact, St. Jude put in $52 million into the project. So it was a bold challenge, though. At the time we started this, the day we started it, only 1 human cancer had been whole genome sequenced. And we said we were going to do 600 in 3 years. The other thing is we hadn't done any next-gen sequencing at St. Jude at that time, and we didn't have the informatics set up at all. We actually didn't even have a bioinformatician at the level we needed to run this. So the goal was the first year, we're going to set up the infrastructure, set up the logistics, and we're going to sequence 50 cancers. The second year, 250 cancers. The third year, 300 cancers. So we're nearing the end of the third year. We're at 475 cancers as of Monday. The data has come in. What I can say is, I had many, many sleepless nights when this project got started. $65 million, I was the head of the project. What if we didn't find anything? What then? What was the purpose? Are we going to be able to actually carry it out? Are we going to be able to interpret the data? Is the quality of the data going to be good enough? Are we picking the right tumors? From my estimation, this has been more successful than I could have possibly dreamed. Every tumor that we've looked at, we have gained new insights into the biology of that tumor. And so this is a figure of about 260 to 300 of the cases, the actual frequency of mutations in individual cancers. And so first I'll show we've published since beginning of January this year. These are the papers that have come out on the Pediatric Cancer Genome Project. So methodology papers, type of ALL, retinoblastoma, diffuse intrinsic pontine glioma, neuroblastoma, medulloblastoma, acute megakaryocytic will be out very shortly. And we have a large number of papers that are either in review right now or in the preparation, with many more coming. So what we're looking at here is individual tumors across here. We've clustered them into leukemias, solid tumor, and brain tumor. And then the individual types of leukemia or solid tumor or brain tumor are here. The different rows then are, these are the mutations, single nucleotide variation encoding regions. We then have single nucleotide variations that are silent. We have tier 2 mutations, which are regulatory regions. We have tier 3, which are non-repetitive regions of the genome. We have tier 4 that are repetitive regions of the genome. And then we have, so what this frequency distribution shows first is that there is marked variability in the number of mutations across the different kinds of pediatric cancer. When we started, people said, well, pediatric cancer is going to be simple. There aren't going to be that many mutations. The patient hasn't been around long enough to develop all of the mutations that occur in an adult cancer. We can see that that's not true, that there are certain kinds of pediatric cancer, this is osteosarcoma, that have more mutations than are almost detected in any of the adult cancers that have been sequenced to date. There are others, though, like infant leukemia, that has almost no mutations. Again, it has that rearrangement of the MLL gene, but very few other mutations. Similarly, retinoblastoma and a few other, here we have low-grade glioma, very few mutations. So marked differences in the frequency of mutations across the different kinds of pediatric cancer. So what I'm going to do is give you a few sort of take-home messages from this kind of sequence analysis with I think, some clinically relevant stories buried within there. So the first is the complexity of the cancer genome. And so this focuses on the first tumor type that we sequenced, and that was a subtype of acute lymphoblastic leukemia that's called early T-cell precursor acute lymphoblastic leukemia. So this is a kind of leukemia that was only discovered in 2009 by Daria Campana at our institution and by a number of other investigators around the world independently. So in pediatric acute lymphoblastic leukemia, about 85% are B lineage acute lymphoblastic leukemia, about 15% are T lineage acute lymphoblastic leukemia, and a rare subtype of T lineage acute lymphoblastic leukemia is this, ETP. But these are the bad actors. Most of the failure is actually in the early T progenitor ALL. And as I said, we didn't even know it existed in 2009. But in 2009, really doing flow cytometry, looking carefully at those cases, it was realized that this is a kind of leukemia that has a T-cell phenotype, but it's different than other T cells. It has low expression of certain typical T-cell markers, and it has aberrant expression of certain myeloid markers and stem cell markers. And this phenotype was reminiscent of a mouse thymic precursor cell called a double negative 1. And we knew from the biology of the mouse that this double negative 1 cell was able to differentiate into T cells, but it also retained some capacity to differentiate into myeloid cells. So a new subtype of acute lymphoblastic leukemia, a bad actor. We want to learn something about it so that we can better understand the disease, and maybe in the genomic analysis there'll be some new therapeutic targets. So the design of our experiment was to do whole genome sequencing, the entire genome of 12 cases, a very small number of cases. Should point out that when we designed the experiment, the overall Pediatric Cancer Genome Project, you know, in these kinds of projects, you talk to statisticians and you say, okay, how many cases do I need to sequence to have a certain power to detect any mutation that's present at a certain frequency? So do I need 500 tumors sequenced to detect a mutation that occurs at a 5% frequency with an 80% power to detect that? And that's sort of what the statisticians said. And we said, well, we're going to sequence 600. And they're saying, well, you need at least 100 of any different subtype. And, you know, so then we sort of looked at them, we threw them out of the room, And then we said, what do we really want to do? What are the questions we want to ask? What's the goal of that first year of getting 50 tumors sequenced? That's all we're going to sequence. And we decided we were going to do small pilot studies, and we were going to get everybody involved, the brain tumor docs, the leukemia docs, and the solid tumor docs. And we're going to get them in the same room, and we're going to get them talking to each other, selecting the tumors, interpreting the data, and understanding the results. Probably one of the major accomplishments of this has been the unbelievable interaction across brain tumor, leukemia, solid tumor, and basic science that this project has generated. So we did 12 whole genomes. I should say that the amount of data we're generating from this pediatric cancer genome project, if you take the data and you type it as type it on a piece of paper, single space, both sides, store those in 4-drawer cabinets, and then start stacking those 4-drawer cabinets, the amount of data that we'll generate over the 3 years would fill the basketball arena here on campus 30 times over. So it's a massive amount of data, and we had to build the computer infrastructure to really analyze this. So we did the whole genome sequencing. Charles Mulligan is the key investigator leading this particular project within the overall project. The first problem was, how do we analyze the data? So one of the most important things we did in the project was we said we're going to take DNA from St. Jude patients. We're going to have the whole genome sequencing done at the WashU Genome Center, an industrial-level genome center. We are then going to have the data shipped back to St. Jude, and then WashU is going to analyze the data, and St. Jude's going to analyze the data completely independently. And then we're going to get together and ask, what do we find? So we did that on the first patient. And they found 12 mutations. We found 11 mutations. We were both pretty excited. We shared the data. 1 overlapped. Now, this is the best sequencing center in the world, and we're talking less than 3 years ago, and only 1 overlapped. The one that overlapped was RAS. We knew that to begin with. It turns out all the ones they picked out were false positives, and all the ones we picked out were false positive. And all the methods that had been used up to that time were inadequate. And it's really been the 2 groups analyzing independently with different algorithms, continually feeding back and forth, that has helped us to develop algorithms that we think are better than anybody's and are giving us much more accuracy than anyone in detecting mutations. So this is the pipeline we use now. And I won't go through all of it, but you can see novel methods for single nucleotide variation, indel structural variations, copy number alterations. It's all novel methods developed at St. Jude and similarly at WashU. So the analysis is still an ongoing science. There's still a lot of development that needs to be done. So one of the key areas of development has been structural alteration. So we know chromosomal rearrangements are occurring in hematopoietic malignancies, solid tumors, and now in many adult cancers. How do you detect those chromosomal alterations? And it turns out those are the hardest to detect. And so if we just put this up as sort of a model of a structural alteration or chromosomal translocation. So here we have 2 chromosomes joined together. So we're doing fragmented paired end sequencing. So here are the fragments from a library of DNA that would contain this translocation. And what we're doing is just sequencing the ends of these and then lining them up to the consensus DNA sequence. So if you look across that break, there are these kinds of fragments where this end will map to this chromosome, this end will map to that chromosome. And so if you pick those out, you'll say, well, there's likely a break there. That's the way the algorithms were working. But it turns out those generate a very high frequency of false positives from the library construction, false positive from the mapping. There's this other category of fragments where it actually spans the break. Early on, these were only 30 base pair reads, though. That's too short to actually map. Now there are 100 base pair reads. Depending on where the break is, those still may be hard to map. You may not be able to take this and actually map it in these 2 separate areas. So people just ignored it. We said, no, you can't ignore it. This is where you're going to be able to improve the accuracy. And so we developed an algorithm where we essentially look for those kinds of fragments. Once we find one, we then build a contig of all fragments that look like that. We can then take these that we don't know the map, we can build a contig of that, and then we can map that. Then we can come from the other side. Say, in this region, do we have any reads that look like this, where they partially contain it? We can build that contig map. We can go back and forth. And by this, we increase the accuracy tremendously for detecting these. In fact, as part of this paper, we took some published sequencing data from the Sanger Center, one of the other 3 big ones. Broad, WashU, Sanger Center are the major sequencing centers in the world. So they had sequenced the cancer cell line, and they said they detected 34 structural alterations. So we applied our algorithm. We said, well, we detect your 34 structural alterations, but 2 of them are actually in the germline. You missed it in the germline. And there are 50 other structural alterations that you missed. And we took 20 of those at random, and were able to validate 18. So again, less than 2 years ago, the best sequencing centers missing structural alteration. So again, we continue to develop these, but this is a major step forward, and in fact, some of the commercial entities now are using this algorithm because we provide it freely to whoever wants it. So here's how complex some of these structural alterations can look. You can see chromosome 10 to chromosome 4, deletion at material between them. You can see the other end hooks up to this piece, which then hooks up to chromosome 1. You can see this hooks up to chromosome 4, and we can read across these at the base resolution. So we're able to detect now most structural alterations, not all. Some occur in repetitive regions. It's hard to get good sequence across, though. We're going to need longer reads to be able to map those. But we're making great strides in being able to identify these structural alterations. So what did we find in this early T progenitor ALL? So we only sequenced 12 cases, and what we found was a high frequency of mutations that were recurrent across this very small discovery cohort. So we had 3 of the 12 cases with RAS mutations, JAK, PHF6, ETV6. These are genes that were known to be mutated in T lineage ALL. We found a number of other genes that had never previously been shown to be mutated in T lineage ALL, and some of these are therapeutic targets. The JAK kinases clearly are. This turns out to be an activated IL-7 receptor that generates the same signaling pathway. So we went on to look in a much larger cohort now, whole genome in 12, find what lesions we can find. Now look in almost 100 cases and ask what's the frequency of those mutations? Does it vary between early T progenitor and standard T lineage ALL? And this is the results in a heat map form. And so what we can see is in early T progenitor versus non-ETP ALL, you could see that there's a very high frequency of RAS mutations or kinase signaling pathway mutations in ETP versus non-ETP. RAS kinase activating mutations are rare in pediatric ALL, but here 80% end up have that pathway activated, many of those therapeutic targets. We see disruption of genes that play a role in hematopoietic cell development. The PAX5 story, but here it's other genes that are being altered that are dysregulating the differentiation of these cells. And then a surprise was we see mutations in genes that regulate the chromatin status. When we started the project, many people said, okay, you're going to be looking for mutations, but the epigenetics is really the story. What regulates gene expression, the malleable, changeable sort of conformation of the chromosomes and the transcriptional regulatory regions. Here what we're seeing is those genes and their protein products that control the structure of chromosomes and transcriptional regions, they're being mutated. So that was really quite a surprise, and you can see how high the frequency is in ETP versus non-ETP. So, and I'll come back to this at the end. So when we look at those mutations in ETP, they were surprising to us. They were not what we saw in other lymphoid leukemias. They were actually more reminiscent of myeloid leukemias. So the way we had thought about ETP was that it was a T lineage acute lymphoblastic leukemia just at an early stage. We knew it wasn't B cell, but now from a mutation profile, it looks more like a myeloid leukemia. And in fact, in data I'll show you, it turns out this is a stem cell leukemia. We're actually thinking about it wrong. We were treating it as a lymphoid leukemia. We thought it was an early T cell leukemia, and the patients aren't responding. But if we do gene expression profiling on these human leukemias, based on that mutation profile, we ended up going further and doing expression profiling. This was in collaboration with John Dick. If we look at the normal human ETP cells, the expression profile of these leukemias are actually anti-correlated. They're actually much more correlated with normal human stem cells or with leukemia-initiating cells from acute myeloid leukemia. So then in aggregate, the data tells us that Early T progenitor leukemia has this profile of mutations. It's reminiscent of myeloid leukemia. In fact, the expression profile says it's a stem cell leukemia, and that tells us that we're treating it wrong. And we need to modify that treatment and treat more like myeloid leukemia. And in fact, some of them have targetable lesions that one might be able to use specific agents against. So the second story I want to tell you is about transcriptome sequencing. So again, whole genome sequencing, 3 billion base pairs, 30-fold over coverage. It's a massive amount of effort. Transcriptome, let's just look at what's expressed. We do it now in combination, whole genome and transcriptome, to help us understand these chromosomal rearrangements. And I'll illustrate that by this next example. So this one is focused on acute myeloid leukemia, a specific subtype of acute myeloid leukemia. So in 2009, we did copy number of AML, very few copy number alterations compared to ALL. But one particular subtype stood out. That was acute megakaryocytic leukemia. And when we looked at it, there were more copy number alterations in acute megakaryocytic leukemia than the others. So acute megakaryocytic leukemia is a relatively rare form of AML, about 4 to 15% of AML. It can be a very aggressive form. It has an increased incidence of 500x in patients with Down syndrome. We don't understand why. And the patients with Down syndrome that get acute megakaryocytic leukemia do well. If we look at non-Down syndrome AMKL, infants have a translocation. We understand the genetics behind that. But all the other patients, we really don't understand the genetics, and they have a very poor outcome. And so these are the ones with complex chromosomal deletions, amplifications, and translocations. And so we said, why not do transcriptome sequencing on those and see, can we define what these rearrangements are generating? Are they generating something that contributes to the development of the disease? Is it something we can use to help us better diagnose the patients? So what we did was transcriptome on 14 non-Down syndrome acute megakaryocytic leukemias. We did whole genome sequencing on 4 of those, and then we had a recurrency cohort of both pediatric and adult AMKLs. Tanya Gruber is the faculty member who has led these studies. So we thought these rearrangements were generating a fusion product, and we found a fusion product. And it was a novel fusion product not previously identified. And it was a fusion that resulted from a chromosomal inversion that resulted in a fusion of a gene from this end of chromosome 16, GLIAS2, to a fusion of a gene from this end of chromosome, CBFA2T3. And so as a result of this inversion, you generate this fusion protein, or fusion gene that encodes this protein, of CBFA2T3, also known as MTG16, a member of the EDO family, a gene that had previously been implicated in acute myeloid leukemia, but exceedingly rarely altered, and GLIAS2, a transcription factor in the Gli superfamily that's never expressed in hematopoietic cells. So as a result of this fusion, what we're getting is aberrant expression of a Gli transcription factor in the hematopoietic cells at very high levels, and it's almost the entire Gli that's being expressed. This just validates that the translocation is occurring, but the surprise was 7 of those 14 cases actually had this chromosomal rearrangement. So a cryptic inversion leading to a fusion product, and we're seeing in 50% of the cases that we happen to select to be sequenced. Turns out 5 of the other cases that we sequenced also had cryptic chromosomal translocations, but they generated other fusions. These were rare fusions, each only seen in a single case as we look broader. But it turns out these are targeting genes that play a role in megakaryopoiesis or leukemia. So GATA2 is known to play a role in normal differentiation of megakaryocytes. HOXA9 is a known oncogene in acute myeloid leukemia. MN1 FLE, MN1 is the oncogene in acute myeloid leukemia. FLE plays a role in megakaryocyte differentiation. And then we have other HOX members being altered. So almost a goldmine of new discovery in acute megakaryocytic leukemia. So we looked in the recurrency cohort and looked at gene expression profiling and asked, this recurrent fusion, does it identify a distinct subset of these leukemias? And so as we looked at more cases, it turns out this occurs in about 30% of pediatric acute megakaryocytic leukemia. It is never seen in adult acute megakaryocytic leukemia, and it is a very distinct subtype of acute megakaryocytic leukemia and different from other AMLs. Even more importantly, this identifies a bad subtype of acute megakaryocytic leukemia, a horrible outcome in the patients that have this chromosomal translocation. So a prognostic indicator in those patients. So how does this fusion work? How does it contribute to the development of acute megakaryocytic leukemia? And so we've done a number of experiments where we've taken bone marrow cells from the mouse, transduced with retrovirus expressing the fusion product or GLE by itself, plated those in methylcellulose culture, and then serially replated those. In the initial plating, no change in morphology of colonies, no change in the growth or size of those colonies. But as we do that serial replating, these cells continue to grow. Normal cells stop after the first to the second replating, but these, you can see, continue to grow, and they express aberrant megakaryocytic markers and show evidence of megakaryocytic differentiation. So the fusion is actually leading to an enhanced self-renewal capacity and a shift towards megakaryopoiesis. It is insufficient to generate leukemia. I won't show you that data, but it initiates. It by itself doesn't generate. We've gone on, done gene expression profile to try to figure out the downstream pathways. And all I'll show you is that what we see is that we are getting turn on of the hedgehog pathway, but also the BMP pathway. In fact, BMP2 and 4 are overexpressed. We can transduce murine hematopoietic cells and show that it gets overexpressed. We've gone on and developed Drosophila models where if we express this fusion, we actually massively overexpress the BMP homolog in Drosophila and generate a phenotype of a gain of function of BMP. And we've been able to show if we inhibit that pathway, we inhibit that self-renewal capacity. So it's the massive overexpression of a transcription factor, turns on a variety of different pathways that are altering the self-renewal of the cells and contributing to that shift towards megakaryopoietic differentiation, and it identifies a bad subtype of acute megakaryocytic leukemia. So let me just shift in the last few minutes to altered epigenetic regulation, and I'll switch to brain tumor. So this is a kind of brain tumor. It's similar to the class that Ted Kennedy had, a glioblastoma multiforme, but this is a particular subtype seen in pediatric patients. It's called diffuse intrinsic pontine glioma. This occurs in the brainstem of the child. Often you can't biopsy that. The process of biopsying itself would kill the child. So we know nothing about the molecular biology of this. Over the last 5 years or so, an autopsy protocol has been generated to get material at the time of autopsy from these patients so we can take that material and look at the genetics and see if we can understand it. So we were able to do this study. This is led by Susie Baker. And what we ended up sequencing was 7 diffuse intrinsic pontine gliomas. And then you see we have a number of cases, 43 other DIPGs, 36 non-brainstem gliomas for sort of a recurrency cohort. If we detect anything, how often do we detect it in a larger number of cases? The results were just remarkable. What we found was that 78% of the diffuse intrinsic pontine gliomas had mutations in histone H3, 2 of the histone H3 genes, and it was the exact same residue in every case. And the residue was K27, lysine 27, and it turned it from a lysine to a methionine. And this is a key regulatory site in histone H3. When it's trimethylated, it leads to repression. When it's acetylated, it leads to activation. This mutation eliminates the possibility of that occurring. We also detected mutations in K34, which is near the K36 site, and others have shown that this mutation actually leads to an inability for that site to be methylated. So this is the first time that the actual target of epigenetic regulation, the histones itself, were found to be actually mutated in a cancer. And here you can see it's 78%. So here is that C-terminal tail, and what we're getting is a mutation in that lysine right there, a mutation here that affects the lysine right there. So a remarkable result, and also this is not seen in any other pediatric cancers. We've looked at the 450 or so that we've sequenced, and it's not seen in adult cancer. So despite glioblastoma being similar in adults and pediatric, it's a very different disease with a very different pathogenesis. Now, it doesn't give us a way of better diagnosing. It doesn't really help us treat these patients, although it does tell us they have a fundamental dysregulation of their gene transcription through this altered epigenetic site. There are many drug companies developing drugs against epigenetic regulators, so it's possible one of those might have some effect, but there's a long way from here to there. So epigenetic regulators and actually mutations in epigenetic regulators. And the more we look, the more we see. I tell you diffuse intrinsic pontine glioma with the histone mutations, but we have histone erasers, histone writers. Here we have infant acute lymphoblastic leukemia. It never has mutations in epigenetic regulators. But if we take older kids with MLL gene rearrangement, about 30% actually have mutations in epigenetic regulators. Same MLL translocation, one does not require epigenetic mutations. The other one, they seem to play some role in contributing. Why is that? Is that different target cells that are being hit by those translocations? If we look at here, early T progenitor, I told you frequent alterations in this pathway. Here's one I haven't mentioned that came out in Nature several months ago, medulloblastoma, very high frequency of mutations in epigenetic regulators, in particular subtypes of medulloblastoma. So we've started to dig more deep into this, and we've developed a custom array where we can now rapidly look at the 600 or so genes that play a role in epigenetic regulation, and we can look across 1,000+ cases very quickly. And so we've just recently completed this study. What I'll show you, though, is just sort of one tail of that study. And this is another kind of acute myeloid leukemia. This is core binding factor leukemias. This is what I've spent most of my career working on the leukemias that are a result of the 821 translocation or the inversion 16. So this is just a mutation profile across these two subtypes. It includes pediatric and adult cases. What I'm going to focus in on is just this area of that heat map. So it was thought that the 821 affects the AML1 or RUNX1 DNA binding component. The inversion 16 affects this CBF beta part of the heterodimeric transcription factor. So two sides of a coin should be pretty similar. Clinically, they're not that dissimilar, a little bit of difference. Well, it turns out the mutation profiles are completely different. And here we see a high frequency of mutations in chromatin regulators and a very high frequency of mutations in cohesion genes, not previously shown to be mutated in any cancers. The cohesion genes are mutations that are leading to changes, and these are genes, without going into any detail, are likely playing important roles in the interaction of enhancer and promoter to play a role in gene regulation, and we now are detecting mutations in these genes in one subtype of leukemia, the 821 leukemia, but not in inversion 16. Raises lots of questions. Why is that occurring? What's the difference? So let me end with the dirty little secrets. So we can sequence the whole genome, we can analyze it, we can detect everything. Why not just start doing this on patients? It turns out we can detect what's there in the sequence information. Sequence information is not perfect. If we look at the coverage, the relative ability to sequence a gene, it turns out, depending on how much GC content is in the gene, our ability to detect it varies. And so if we look at genes, we just artificially divide genes into 10 exons and then take all genes in the human genome where the GC content is less than 40%. We get pretty good coverage. If you now take those genes where the GC content is 40 to 60, well, the coverage goes down, but the first exon goes down. Obviously, GC content's even higher in that first exon. If we go to even higher GC content, we're not getting coverage of that first exon. In fact, if you look at a gene, here's the retinoblastoma gene. Here's the level of coverage across the different exons. Look at this exon right here. There's no coverage. And so there are parts of the genome, coding parts of the genome, parts that are actually critical, parts where we need to know if there are mutations, and we're not covering them very well. This is a process of developing the libraries. It's the actual reading of the sequence. There's a bias against GC in the detection of the fluorescent. If we do exon sequencing, there's a bias in the capture. So this is a problem as we apply this into the clinic. We're not detecting everything, and somebody needs to critically look, what are we missing? How do we improve the methodology or the analytical pipeline to make sure we don't miss it? So other things, as I briefly mentioned on structural alterations, we have short reads, so some of them we just can't detect. We need to improve the read length. We might be able to get that up to 250 with the methodology in use now, but it'd be much better if we could get it up to 1,000. That's going to take new methodology. The other thing is, if we look at the literature and think about what people are reporting and what they're actually doing on patients, most of what they're reporting is exon sequencing. That's 3% of the genome. Do we really think the other 97% of the genome is not going to be important? What about the mutations in those regulatory regions? We don't even, you notice I haven't mentioned them. We don't even know how to look for them. We don't even know how to find them. You know, they're not going to be a single base change. They're disrupting a region. It might be a structural element of the region. And so that's going to take a lot of work to figure that out. So germline polymorphisms, I won't even touch. We have a long way to go to understand those. So I think cancer genomics is clearly providing valuable insights into the molecular lesions that underlie cancer, and they are generating and insights into the lesions generating therapeutic resistance. I think an important point is that the discovery phase is not over. There are some that say, well, I keep going with cancer genomics. It's going to be diminishing returns. We've gotten all the important insights. No, what we've got is the low-hanging fruit. There is tremendous discovery that needs to be done. The regulatory regions we haven't touched. There are parts of the genome that we're still not able to accurately see. And very few cancers have actually been sequenced at the level to give us that landscape of mutations. So it's an area where a lot of work needs to be done to really give us the kind of databases we need to accurately apply this into the clinic. So let me end with that statement. Not to say that we shouldn't be applying it to the clinic, but what we're doing today in the clinic is not what we're going to be doing 2 years from now or 5 years from now. We need to continue to advance that. So these are the credit to the people who've really done the work. It really, is just a very small list. There are over 150 individuals at St. Jude dedicated to the Pediatric Cancer Genome Project, over 200 individuals at WashU dedicated to the project. We are now designing phase 2 of the project and a big effort in clinical genomics, and we'll get those started sometime in the next several months. So it'll be ongoing work over the next several years. And then I have to give credit to St. Jude Children's Research Hospital. As I said, $52 million of this $65 million bill was paid by our fundraisers, but that's why they raise money. I know they raise money in this city, they raise money in every city in the United States, but us as leaders of that institution, we see our job to do things that need to be done in the realm of pediatric catastrophic disease, to generate that data and then share it rapidly with the scientific community so that they can use it to advance their research. And so the pediatric cancer genome data, we've put it on a website, even the unpublished data, and are making it available for researchers. All they have to do is apply for it. We approve all applications as long as it's from legitimate organizations, and we allow them to have the data. They can't use it in a publication for a period of 9 months if it's unpublished, but we give them access to it so that they can use it. And I should say the federal government project has an 18-month moratorium, and we felt that was too long. Ours is a 9-month. So let me end with that. I do apologize for going over, but happy to take any questions if there are any.